Hey guys, so as you already know, we already started this topic in class. I'm just going to kind of go through the slides that we've already gone through because I want you to have the entire video just in case you ever need to like re-watch it or listen to it or whatever. But I'm going to do it fairly quick and short and concise because you already have notes on that. You already got all the stories and that kind of thing. On the slides that we haven't already covered, then I'll go slower and the video format will be the way it normally is. So here we go. So there are two kinds of reproduction, asexual and sexual. Asexual happens through mitosis. And in mitosis, we are producing identical daughter cells that have the same information. So these cells are exact copies of your original cell. When we say they have the same information, we mean that they have not only the same number of chromosomes, but all of their chromosomes carry the same genetic information, the same code in the exact same way. No changes. Now, most organisms go through asexual reproduction through a process called binary fission. So if you look at this picture right here, this will show you an amoeba cell, and that amoeba cell, you can tell that it used to be one big cell and now it's splitting down the middle, and that's binary fission. Another way of doing asexual reproduction is through a process called budding. And if you look at these two pictures, they both show budding. It's almost like you're taking a section of your body and you're growing a mini-me off of that section. So what I'm doing here is I'm just circling where those buds are. Now once the cell, or not the cell, but yeah, once the section, that little part that you're growing gets big enough and can handle on its own, then it severs the connection with the main organism and it becomes its own free-floating, living, breathing organism. Now, single-celled eukaryotes like yeast, which is what these are, and protists, like amoeba up here, they all do asexual reproduction. You can also find some multicellular eukaryotes that will do asexual reproduction, but they're very simple organisms. So this is an example of a hydra, and it too does asexual reproduction, but it's a fairly simple organism. So what are the disadvantages? Well, you're making the exact same thing in the exact same way. There's no kind of genetic variation whatsoever, and that can't be good, at least not long term. What are some of the advantages? This is a quick process. It happens really fast. You don't have to do many steps and, change up too many things. So it's easy and quick to do. And when you think about using asexual reproduction for growth, then you want to be able to grow fast and repair fast. Now, when you become a complex organism, you can't just do mitosis anymore, especially when we need to reproduce and extend the species. Now, you still do mitosis for things like healing cuts and, broken bones and stuff like that. But when you're making a new organism, we have to go through a different process. To create a new organism, it means that we have to join a sperm and an egg together. So that means we have to make these cells. This is an egg cell and a sperm cell. We don't make those cells by mitosis because if we did, we would have way too many chromosomes. We'd have an egg cell with 46 and a sperm cell with 46 because remember, mitosis is all about keeping things the same. So this zygote would have 92 chromosomes, way too many. Right, so real quick, this is a picture of what human chromosomes look like, and when it's laid out in this order where we pair them up, we call it a karyotype. So this particular karyotype is female. You notice that we have 46 total chromosomes in terms of individuals, but we have 23 pairs of chromosomes. We know it's a girl because 2 X chromosomes. All right, and here's the boy, same 46 chromosomes, same 23 pairs. We know it's a boy because there's 1 X and 1 Y. So, homologous chromosomes are paired chromosomes, meaning they've replicated and now you have 2 of them, okay? You have homologous chromosomes because you get 1 chromosome from mom and another chromosome from dad. Now, the word homologous, homo means same, logos means information, so homologous means same information. These paired chromosomes are carrying the same inherited characteristics. They're carrying matching genes, if you want to think of it that way. So like right here, this could be your gene for your hair color on dad, and here's your gene for your hair color on mom. Okay, so you get two two versions. Now real quick, I want to show you, right here, we have what we call single-stranded homologous chromosomes. So this means that these guys exist before replication. Okay, whereas when we have our double-stranded, it means that replication has taken place. Okay, so just be aware, before replication, after replication. So how are we going to go about making these sperm cells and egg cells? Well, we know that our organism has to have total 46 chromosomes. And since we have two separate cells, it means that each cell has to carry 23 chromosomes. Now, please note I'm not saying pairs. I'm talking about actual individuals. So we have to take this 46 and somehow reduce it down to 23. We have to half the number, in other words. So in meiosis, that's what we do. We find a way to take our 46 chromosomes down to 23 in the egg and 23 in the sperm. And then when those cells fertilize, when they fuse or join, they reblend all of their DNA, and we end back up with a zygote that has 46 chromosomes. So every organism goes through what we call alternating stages of life. And we haven't really used these words before, but we'll use them now. We're going to talk about the words haploid and diploid, at least we did in class. And again, like I said, this is just a refresher. So these words refer to how many chromosomes we have at different periods of time. Haploid, if I think of it as haploid, kind of sounds like half, or, you know, sorry, still can't write with this. I think of haploid as being half, and I think of diploid, I think specifically of the di, which is 2. So 2 times your half. That's how I remember that. Hopefully this helps you. So we know that chromosome numbers must be reduced. Your diploid chromosome number represents your total number of chromosomes that you're going to have in all of your somatic cells, which for a human being is going to be 46 chromosomes. Your haploid number represents half of that. How many you're only going to have in those gametes or those sex cells that you're making, which in this case it's represented by the number 23. Another way we can depict diploid and haploid is by using 2n and n. So in this case, n would be 23 and 2n would be 2 times 23, which is 46. So again, don't forget, the point of meiosis is to reduce your chromosome number. You're trying to make gametes. You want, when fertilization here takes place, that you're going to get a zygote with 46 chromosomes, not 92 chromosomes. So meiosis reduces the chromosome number, and fertilization restores it. Meiosis takes you from diploid to haploid. Okay? So diploid here, haploid there. And then fertilization takes you from haploid back to diploid. So I hope that's clear. Okay, so this is just another copy, like a cartoony version that goes over the same thing that that picture went over. So I'm not going to talk about this in detail. It's pretty much the same thing. It's just represented in a more simplistic form. Okay, so in meiosis, we are reducing. It's all about cutting that diploid number in half. So it's a special cell division, but it's only used for sexual reproduction. Mitosis is used for asexual reproduction, but meiosis is only used for sexual reproduction. The result is taking our diploid number, our 2n number, and reducing it to our haploid number, or our n number. So we're going from 2 to half. At the end, we're going to make sperm cells if you're a boy, and egg cells if you're a girl. Now, hopefully you've noticed a trend that in biology, we start off with the simpler process, and then we make some changes and some tweaks here and there, but we keep pretty much everything the same. So mitosis was our first reproductive format, and, meiosis, we just kind of evolved from mitosis. So the stages that we talked about for a little bit are going to be very similar. The method of separating the chromosomes and spindle fibers and all of that, those are going to be similar as well. But the process and the results are very different, so make sure you don't confuse the two. All right, so let's overview real quick. Now, initially, meiosis is going to look just like mitosis, with the exception of we're dealing with pairs of chromosomes here. So I hope you notice that. If you look here, this is one pair of chromosomes, and here's another pair of chromosomes. Don't forget, one pair from mom, one chromosome from mom, one chromosome from dad. So that's how we end up with that homologous pair. Now, at first glance, the first part of meiosis looks, it's the same process, except that we're dealing with two sets of chromosomes. There is one thing that takes place. I want you to pay attention to this right here. In prophase 1, I want you to notice how it's almost like they link arms, and then after they pull away, we have a little bit of genetic variation, and we'll talk about this more in depth in a little bit. But I want you to make note of this event right here. Now, this first part is almost like meiosis. Sorry, it's almost like mitosis. And the result is going to be two daughter cells. Okay? Now we're going to take those cells, and each of them is going to go through another set of division. So you have division here and a division there. The result is going to produce two sets of cells in each division. So you'll have two new cells here and two new cells there for a total of 4 cells, which means that we're going to reduce our chromosome number from the original to whatever that half is. Okay, so up here, our original chromosome number is 4. 1, 2, 3, and 4. When we end meiosis, okay, look at us right here. When we end meiosis, and we start dividing all over again, we are now 2. 1, 2. So when we go through the entire process to end up with those 4 cells at the end, to end up with the 4 cells at the end of the process, we are still 2. 1, 2. 1, 2. Okay, so this is how we go from 4 to 2. This is how we have our chromosome number. Now you should notice that the names of the cells, of the phases, are the same. There's interphase and prophase and metaphase and anaphase, but we have them for the first time, and then we have them again for the second time. Okay, so in meiosis, what you're going to realize is that we have a double division taking place. The important part we're just going to pay attention to on this particular slide. Remember, before we can divide, we have to replicate. So we're still going to have interphase. Our cell's still going to grow. We're still going to check for nutrients and growth factors and all that stuff. And once everything is okay, we will go ahead and we will replicate, which is how we get those homologous pairs. The first division, or what we call meiosis 1, is going to separate the homologous pairs. Okay, because when you start off, you have mommy and daddy's chromosomes together, mommy and daddy's chromosomes together. When we go through the first part of meiosis, we're going to separate dad's chromosomes from mom's chromosomes from dad's chromosomes. Okay, so like this might be mom's chromosome, that might be dad's. This one is dad's, that one is mom's. Okay, so we separate them out. Now when we undergo the second division of meiosis, we're going to separate those chromatids that we talked about. Okay, so I hope that's clear. Division number 1, we separate homologous pairs. Division number 2, we separate sister chromatids. So in the first step of meiosis, like I said, we're going to go ahead and duplicate DNA. Why? Because this process evolved after mitosis. In mitosis, we replicate DNA. It's convenient to use all the parts and pieces that we still have. But remember, these phases are not going to be phases of meiosis. Okay, so they'll be interphase of meiosis, just like how we have interphase of mitosis, or interphase leading into mitosis. So we'll go from being single-stranded, like we are here, to double-stranded there. Note our chromosome number doesn't change. We didn't go from 6 chromosomes to 12 chromosomes. We went from 6 single-stranded chromosomes to 6 double-stranded chromosomes. It's important that you note that. So like I said, in meiosis 1, it's a division of homologous pairs. Now in prophase, something special happens. The chromosomes arrange themselves into what we call a tetrad, and it's an arrangement of 2 homologous pairs like you see here on the screen. Now I want you to notice how, and I call them the arms and the legs just to keep it clear. I'm going to switch to my laser. I want you to notice how this red chromosome has crossed its arm with this blue chromosome. Then it's gone ahead and crossed its leg with this blue chromosome. Note it only happens on the inner chromatids, not the outer chromatids. Okay? This crossing, this crossing like this is called a synapsis. Now, this is really important. Why is it so important? This is where you're going to start to get some of that genetic variation that sexual reproduction is known for. Because when these chromosomes cross their arms up here and then their legs down here, they exchange this portion that's crossed. And it'll switch which chromosome it's found on. So this chromosome is going to become mostly red with a little bit of blue. And this chromosome is going to become mostly blue with a little bit of red. It's almost like I linked arms with someone, and when I unlinked, they have my glove, and I have their glove, and they have my sock, and I have their sock. We're exchanging information. Okay? Now, in the second division of meiosis, we don't have homologous pairs anymore. If you look at our cells, we just have replicated chromosomes. We don't have homologous pairs. So now what we're going to do is we're going to separate these chromatids. All right, we're just going to split them right down the middle. This should look like mitosis the most, okay, because that's what we do in mitosis. We separate sister chromatids, okay? Please note, sorry, I forgot to mention this. Please note that at the beginning of mitosis 2, we are already haploid, okay? So, just to go back over so that you have it, the first division in meiosis separates our homologous pairs. It separates mom's chromosome from dad's chromosome. This is where we reduce our chromosome number. We go from 4 to 2, or we go from 8 to 4, or whatever the chromosome number is. In the second mitotic division, we separate sister chromatids. So we're not changing the number. We're not going from like haploid to something else. We're staying haploid. We're going from haploid to haploid, if that makes sense. Because we're just splitting the chromosome in half. This second division is the most like regular mitosis. Right, here are just some pictures of cells going through mitosis 1 and mitosis 2, just so you can see how they pair up. Now, let's go back and talk about that genetic variation that I mentioned before. And remember I said when your chromosomes arrange themselves and the pairs cross arms and legs, this happens during prophase 1. And we have an intertwining of your chromatids, the inner chromatids, this one here and that one there. This is called a tetrad, this arrangement. What happens is that the homologous pairs swap pieces, like actual sections of chromosome. The DNA breaks off completely off of one chromosome and reattaches itself to the other chromosome. So when they move apart, this is what you end up with. You end up with a mostly red chromosome with small sections of blue, and a mostly blue chromosome with small sections of red. What we've just done is we've changed up our DNA a little bit. We have introduced some variation right here. And that is what sexual reproduction is known for. It's the introduction of new genetic information, material. We've switched things around. Okay, so crossing over happens in 3 steps. There is the actual crossover, then there's the breakage of DNA off of one chromosome, and then the refusing of DNA in onto the other chromosome, and not refusing as in I don't want to do it, but refusing as in I'm reattaching myself. So what this gives us is a new combination of traits. We have traits on either chromosome, on mom and dad's chromosome, that we didn't have before. And now when we separate into sister chromatids, this particular chromosome is going to have some information that this chromosome doesn't have. And same thing here. This one is going to have information that that one doesn't have. So that's the advantage. We are introducing genetic variation into our organism. Okay, this particular slide just compares mitosis with meiosis. It's a very good one that you might want to pause and just look at some stuff, maybe write some notes down. We've talked about all of this already, so I'm not going to go over it. Like, you already got it. All right, and then here are the words. So same thing. We're comparing mitosis to meiosis. So in mitosis, it's one division. We're going to make daughter cells that are genetically identical to the parent cell. We're producing 2 cells total, and we keep the diploid number. We go from 2n to 2n. These cells are used for growth and repair only, and no crossing over takes place. In meiosis, it's 2 separate divisions. We still make daughter cells, but they are genetically different from the parent, and they produce 4 cells total. We have our diploid number, so we go from 2n to n, and we produce gametes or sex cells. Crossing over does take place. This is used for the making of a new life, sexual reproduction. All right, so just again to put it all in perspective, meiosis allows us to take our diploid condition down to the haploid condition, thereby allowing us to make gametes in the form of eggs if you're girls and sperm if you're boys. The fusing of these egg cells and sperm cells is called fertilization, and it allows us to reinstitute, for lack of a better term, the diploid condition. This leads to a zygote, which over time, with lots of care, can produce an actual living organism. This organism's body cells are all going to reproduce by mitosis. Development is controlled by mitosis. Okay, so let's go over the value of sexual reproduction. Sexual reproduction produces genetic variation. It's what we call genetic recombination. We're moving things around and recombining them in new and different ways. Now, because chromosomes assort independently, and that just means that any chromosome has a chance of moving into any daughter cell, it's all random, okay? Because it's all based on how they line up during metaphase 1. Crossing over is also going to give you some of that genetic difference because you're going to have a mixing of alleles on chromosomes that are actually homologous. When those chromosomes split into chromatids, we're going to have one completely red chromatid and one red and blue chromatid. These two are going to be different genetically from each other. Fertilization is random. It just means that we don't know which sperm cell is going to fertilize which egg cell. We have no clue what particular genes or alleles or traits are on those individual chromosomes in that particular cell. This is the process that drives evolution. It provides the raw material for natural selection in the form of variation. All right, so just to put it in perspective, in humans, it is possible to have 2 to the 23 different combinations of gametes. So who knows what you're going to end up with? It's all spontaneous, okay? Because of crossing over, we're going to get combinations that are new and different on each chromosome. It creates an infinite variety in terms of gametes, lots of differences, and lots of areas that crossing over can take place. Now, we don't know which sperm cell and egg cell are going to meet up. Any two parents could produce a zygote with over 70 trillion possible diploid combinations. That's why, unless you have some very distinctive features that stand out, couples don't really necessarily give birth to identical children. The children aren't identical, unless they are actually identical twins, but the children aren't identical to each other, and they're not identical to their parents either. Lots of variation. You can look at one whole family and not necessarily know if they're all closely related, just because there's so much differences. And that's all because of how these sperm cells and egg cells combine. Right, so just to give you some examples, here are the Jonas brothers, who, I mean, they don't look hugely different from each other. Actually, these two resemble the most out of the four of them, but they're four very, I mean, they're differences, obviously, but they're all genetically related. Here is Martin Sheen and Charlie Sheen, and here's their dad. I mean, yeah, you can see some similarities, but they're not identical, okay? Here are the Baldwin brothers. Again, not identical. All came from the same mom and dad. But there's definitely some variation in between. So sperm cells are made through a process called spermatogenesis. It is continuous and prolific, which just means that it happens throughout the duration of the male's life. And they produce millions and millions and millions and millions of cells. I mean, that's just how it goes. They're really, really tiny. So real quick, let's look at some anatomy. Sperm are produced in the testes, and this is what the testes look like on the inside, like if you did a cross-section. They're stored in the epididymis, and then when they're ejaculated, they pass through this tubular pathway called the vas deferens until they exit the body. Now, if you could zoom in on a section of the seminiferous tubules, which is where sperm cells are actually made, this is what you would see. And this is the sperm cell going through meiosis 1 and meiosis 2. They all start off as a germ cell. The germ cell is your diploid cell. And then they change form, and they go through meiosis, and then they go through meiosis 1, and then meiosis 2. Each new phase has a name. So, for example, from germ cells, we become primary spermatocytes. Then from primary spermatocytes, we become secondary spermatocytes. Then we become spermatids. And eventually, the actual sperm cell is known as spermatozoa. So egg production takes place through a process called oogenesis, and it's a little bit different from spermatogenesis in the sense that women are born with all of the eggs they need. Those eggs are held in the ovaries, and while you are still sexually immature, the eggs are halted before they get to anaphase 1 of meiosis 1. Now, upon sexual maturation, meiosis 1 is completed, but meiosis 2 is not completed until after fertilization takes place, until after we fuse an egg cell with a sperm cell. The result is going to be one egg and two smaller reduced bodies that we call polar bodies. Right, so this is what this diagram showcases. Feel free to draw it, look at it, take notes over it, whatever. Okay, now oogenesis does happen in the ovary, and it's almost a circular pathway, and this just kind of showcases all the different names and all the different ways that the egg is going to look before it's finally released, not reduced, but released, well, reduced as well, and sent into the fallopian tubes where it's going to be delivered to the uterus. Okay, now, not all organisms use haploid and diploid stages in the same way. For our life cycle, our dominant phase is our diploid phase, our 2N condition. But in other species, which one is dominant is going to differ depending on what the species is. Like, for example, in some plants, you have an equal division where you're equally diploid and equally haploid. For this, you're pretty much in the haploid or diploid condition for the same amount of time. It's like your life cycle is exactly halved. In half of it, you're diploid, and the other half, you're haploid. Excuse me. Whereas in organisms like Okay, sorry about that. Like I was saying, in organisms that are simpler, like algae, for example, you're haploid most of your life, and you're only diploid for a very small amount of time. So it just depends. But do realize that they're all going to alternate between being haploid and diploid. It just depends on which ones. It just differs based on which one's dominant. You have to alternate your condition so that you can go through sexual reproduction. So anything that does sexual reproduction is going to spend time going back and forth between haploid and diploid. Okay, so hopefully this was fairly clear. Feel free to ask me questions in class, anything that you want me to clarify or go over. Have a good night.